Hi guys, this is the screencast on Chapter 18 of our Campbell Biology book, The Regulation of Gene Expression. So now that we have learned about the central dogma, and we've learned about how DNA can copy itself, and then how that code can be transcribed and then translated into a protein, now we're going to learn about how we regulate that. And this field has just exploded in the past several years, and we're learning so much. It's really hard for us as teachers to keep up with all this, especially biology teachers. So here we go, we'll get started. So we're going to start off talking first about how bacteria do this. So you know that bacteria have very simple systems, and so what you're going to see in how bacteria control their gene expression is going to be very different than what we see with eukaryotes. There's some similarities, but basically it's a simpler system. So basically, when you think about bacteria, and you think about bacteria being the oldest living thing that we have on Earth, they've been around for literally billions of years. One of the things that we know that makes bacteria very successful is that they can respond to environmental changes. And they need a very quick response to be able to turn genes on or off based on what is happening in their environment. So back in 1961, a guy named Monod came up with this model of how bacteria control their gene expression, and he called it the Operon model. So remember that we have already learned that in a metabolic pathway, when you have different enzymes that are going to catalyze certain parts of that metabolic pathway to make something, one of the ways that any living cell can control that is by the regulation of the enzyme activity with negative feedback. So we're going to talk about the fact that bacteria need to produce tryptophan, and there are several different enzymes in this pathway that bacteria use to produce tryptophan. If they have a whole bunch of tryptophan, they don't want to keep cranking it out, so they can have a negative feedback that is a very quick response to alter this enzyme or one of the enzymes in the pathways to slow things down. Another way that bacteria can control its metabolic pathways is a little bit slower, but they can actually regulate the making of those enzymes. And that's what we're going to talk about right now. So this pathway, the regulation of the enzyme production, actually making those enzymes with protein synthesis, is a little bit slower response, but it also works as well. So that's what we're going to talk about now. So the basic concept of an operon is this idea that there is a switch, and then downstream from this switch are some structural genes. And those genes are going to code for specific proteins. And so the regulatory switch is a piece of the RNA. So this is all on the, this is all on the DNA. This is all on the DNA. The operator is a switch, and it's usually positioned within something called the promoter. The promoter is where the RNA polymerase is going to sit when this piece of DNA gets transcribed. And so sometimes upstream from the promoter and the operator is a repressor gene. So an operon is a stretch of DNA that includes this regulatory region, the promoter, which we say is like a doorknob. That is where the RNA polymerase is going to sit. The operator, which is going to have, which may or may not have a protein that sits on it that can repress that, and then we have these structural genes that are downstream. So the operon is the regulatory region plus the structural genes, with a repressor gene that makes another protein upstream. Okay. So the operon can be switched off by a protein called a repressor. So this regulatory gene that was upstream from the promoter doorknob, which is where the RNA polymerase sits, this regulatory gene can sometimes make a protein called a repressor protein. And when that repressor protein is present, it can sit on the operator region. And when it does that, it blocks the promoter region. And so the RNA polymerase cannot sit there and it cannot transcribe these structural genes that are downstream. Now, in this case, the repressor in red that was made was actually inactive. It needed a co-repressor to be able to bond to it, and now it's a fully functioning repressor, and that repressor can sit on the operator region. So this is giving you foreshadowing into the level of how bacteria can control whether genes get turned on or turned off. So guys, there are basically two types of operons. There are inducible operons and repressor operons. So inducible operons are going to be operons that are normally not turned, they're normally off. And so the effect of an inducible, it can be induced or turned on. So the lac operon is a great example of that. A repressible operon is an operon that is always turned on and can be turned off. An example of that is the trip operon. So let me just give you a little bit of an example here. So let's start with trip first. So tryptophan is an amino acid that that bacteria are going to want to make. They need to make enzymes to make that. And that's something that they generally keep turned on. They keep making tryptophan, keep making tryptophan, keep making tryptophan. But when the amount of tryptophan builds up, there's got to be a way to turn that off. And so there is a way to repress it. So the tryptophan operon is normally on, but then can be turned off, right, in bacteria. Remember, this is all in bacteria. So let's talk about the lac operon. So bacteria would rather use glucose as their primary fuel, but they can also use lactose. And when lactose, which is a disaccharide, needs some enzymes to be able to cut it into two monosaccharides, galactose and glucose. And so when conditions are right, if lactose is available, then the bacteria would want to turn on the genes that have the enzymes to break down lactose. So lactose is an inducible operon, and tryptophan is a repressible operon. So the first one we're going to look at is the trip operon. All right, so here we go. Let's just take a look at this. The actual operon is right here. Okay, so we have a promoter here, and then we have the operator that sits right here, and then we have our regular structural genes down here. These guys are coding for polypeptides that are going to make up the enzyme that helps the bacteria make tryptophan. All right, so upstream we are going to have a regulatory gene. It has its own little promoter, but this is a regulatory gene. And the regulatory gene can make a piece of RNA, and that messenger RNA then gets made into a protein. But the regular way that this protein is made is in its inactive form. So it will not go over here and sit on the operator. But this is when tryptophan is absent. So the operator is on. Bacteria is making these enzymes. Here's my RNA polymerase. It's going down. It is transcribing. Here's my messenger RNA. And it is making these polypeptides that are going to make up the enzymes that make tryptophan possible. And so now you can imagine tryptophan building up in the bacteria. So as tryptophan builds up, the bacteria needs to have a way to say, okay, we have enough tryptophan. And so the tryptophan is going to be a co-repressor. It's going to fit on this inactive protein. And now it is an active repressor. And that active repressor is going to bond with the operator or onto the operator site, and now my RNA polymerase cannot bond. And so no more of these structural enzymes are going to be made because the cell has enough. If the amount of tryptophan goes down, then it's going to, these proteins are not going to have the co-repressor, and it will fall into the inactive position, and then it will go back on again. In the case of the lac operon, remember, the lac operon is normally turned off because the bacterial cell would rather use glucose. And so when glucose is not present and when glucose is present, then it doesn't need to use lactose. It would rather use glucose. So the normal position of the lac operon is off. All right, so the regulator, the repressor protein is being made from the regulatory gene. Here's my piece of messenger RNA to make this protein. It is already synthesized in its active form. And so this active repressor is going to sit on top of the operator. And now my RNA polymerase cannot bind to the promoter. Remember, that's the doorknob. So that repressor kind of blocks it. I'm not going to synthesize any of these enzymes, these structural enzymes that are downstream. So when glucose is not present and the bacteria says, okay, I will go ahead and metabolize lactose because I have to. What happens is that when lactose is present and the cell can use that, there is another form of lactose called allolactose. And allolactose is always present when lactose is present. And they're isomers, if you remember that term, isomers. So what happens is this allolactose bonds to the repressor protein, making it inactive. Remember, this is only when lactose is present. So now that inactive repressor falls off the operator, the RNA polymerase now can fit onto the promoter, and now it's going to start transcribing. It's going to start transcribing these structural genes. And here's my piece of messenger RNA for those genes. And these genes are going to be coding for enzymes that are going to break down lactose. And here's one right here. Beta-galactosidase is one of the enzymes that's needed to break down lactose. So those are the way that operators work. Now, just to remind you about the difference between anabolic and catabolic pathways. Anabolic pathways are building. So they're pathways where you take small molecules and you assemble them into large ones. Energy is needed. And that's where we're building these. A catabolic pathway takes a large molecule and breaks it down into smaller ones. So I always tell my students, anabolic building, catabolic destruction. It's not really destruction, but it's the breaking down of a large molecule into a smaller one, and energy is usually released. So here is something to kind of keep in mind. Inducible enzymes are usually functioning in catabolic pathways. Remember, the lactose operon or the lac operon is a catabolic pathway because lactose is being broken down. So it is an inducible enzyme. It's normally turned off and turned on when it's needed. Repressible enzymes are usually functioning in anabolic pathways. Their synthesis is repressed by high levels of the end product. What does that mean? So an anabolic pathway is when the cell is building something. In this case, like when it's building tryptophan. As tryptophan, which is the end product, builds up, you don't need that product to be made anymore. And so then you can turn off that operon, and that is repression. So both the trip and the lac operons are negative control of genes because their operons are switched off by active forms of the repressor. Okay, so keep that in mind. It is a way of, you're either turning them on or you're turning them off, but there is a switch that is that little repressor protein that is either fits on the operator or it doesn't, and that's a negative control of genes. As it turns out, we also have positive gene regulation. Now, in positive gene regulation, what happens is that we have this protein called Cap, which we'll talk more about it in just a minute. If Cap is not activated, then RNA polymerase can fit on the promoter and there's nothing blocking the operator, and so it's going to go through transcription and then translation of these structural genes. But in the presence of cyclic AMP, that usually means that there is lots of cyclic AMP hanging around, and the cell is going to be in need of some ATP. It's going to need, in this case, it's going to need some lactose to be able to be broken down so it can have glucose, so it can make some ATPs. But anyway, what happens is with cyclic AMP, it bonds to this Cap protein and it kind of accelerates the RNA polymerase process. So you see this big arrow here means more transcription. And so that's an example of positive gene regulation, which actually speeds it up. So this Cap protein is a catabolic activator protein. And as we've just said before, when glucose is scarce, then Cap is activated and it bonds with cyclic AMP. Cyclic AMP, remember, is one of the precursors to ATP. And so when the cell needs some more ATP, then this is going to be activated and it is going to help to speed up the transcription on the lac operon, which is going to make those enzymes to break down lactose, and then the cell can have the glucose because these enzymes in the lac operon break down lactose into glucose, and glucose is then burned by the cell to make some ATPs. So that is an example of the positive gene regulation. So here's just an example of what I just said a few minutes ago about how the presence of high levels of cyclic AMP can activate this Cap. It's going to sit on the promoter and actually enhance RNA polymerase binding and transcription, and that is something that is going to help little bacterial cells. So guys, that's an example of the Operon system. That is the primary way that bacteria can regulate their genes. In eukaryotic gene expression, it's a little bit different because we're going to see that gene expression can be regulated at many different stages. And this is a very important thing to understand in how cells specialize, because when you have a bacteria, that's just one cell. That one cell does everything. But in multicellular organisms, which most eukaryotic organisms are, each cell has become specialized, right? And that leads to something called cell differentiation. And cell differentiation is when some genes are turned on and some genes are not turned on in specific cells. What genes are turned on allows that cell to become the cell that it's supposed to be. A cell is a specific cell because of the proteins it makes. So I tell my students that you can think of all the DNA in a eukaryotic cell as being like a CD, a music disc. Okay? And so when you were a zygote, when your egg and sperm formed to make you got a CD. And on that CD were all the tracks, all the DNA that you're ever going to have. And that cell, that zygote, goes through mitosis. And so every single cell in your body has the full CD. It has all of the tracks. It has all of the genes that you could ever want. But here's the difference. What happens is the cells begin to differentiate. They begin to only play certain tracks. All right, so a nerve cell, even though it has the whole CD of genes in its cell, only plays the tracks that have to do with being a neuron. So it's only going to turn on the genes for proteins that a neuron needs. These red blood cells only are going to play the particular tracks on the DNA that make the proteins needed to be a red blood cells. Here are muscle cells. They're again only going to make those muscle cells are muscle cells because the genes on the CD are turned on to make muscle proteins, and that is how cells differentiate. So there are many different ways, many different levels of control that a eukaryotic cell can control protein synthesis. And remember, a cell is what its proteins are. And so that's a really important concept. So one way that the cell can control is whether or not the DNA is readable or not. And we call this chromatin remodeling. And we learned a little bit about this before. So we can methylate the DNA, we can acetylate the histones, and that causes the chromatin to change to let the genes be turned on or turned off. We can control at the transcription phase. We can control RNA processing. So after the messenger RNA is already transcribed, we can edit that messenger RNA. And then after the messenger RNA, the mature messenger RNA leaves through the nuclear pores to go out into the cytoplasm, we can still control protein synthesis. We can control at the translation level. We can degrade the messenger RNA before it ever gets to a ribosome. After a protein, after a polypeptide is synthesized, we can modify that polypeptide by activating it, folding it, transporting it, and then we can degrade the active protein itself. So there's many different levels that a eukaryotic cell has. Now this is a diagram directly from your book. I just like this other picture better because it showed them all on one specific diagram. But if you look in your textbook, as you go through the different stages of where the protein synthesis can be controlled, you will see that this particular diagram is highlighted as to what part of the text is talking about which way that the eukaryotic cell is controlling its gene expression. So controlling its protein synthesis. So I think that's a good thing to keep in mind. So we're going to talk very quickly about, this is a review for us, about this idea of chemically putting some tags on either the histones or the DNA. Remember that highly packed heterochromatin is not usually expressed because it cannot be transcribed, and euchromatin is the more relaxed, and that is what happens usually when we acetylate histones. So we can acetylate our histones, the histone tails. These are the proteins that the DNA is coiled up on, and those can relax, and that is one way that we can control protein synthesis. Remember that these tags, these methyl tags and acetyl tags, can actually be inherited. That is called epigenetic inheritance. These are the tags, and they can be these tags can be influenced by the environment. They can be influenced by behavior, nurturing mothers versus not, the abundance of food or not, and certain chemicals in the environment. So that we did talk about that last chapter, but just a reminder that that is an example of controlling protein synthesis. So if we have our methylated DNA that is coiled up into heterochromatin, we're not going to have any genes turned on. I like this picture because it shows a big picture kind of frame. So I've got in my nucleus my heterochromatin, and I've got it relaxed into my euchromatin here. So I could have transcription occurring. Here is a picture of my, my messenger RNA, and I have my promoter region here, and I have my exons and introns. Remember, we have talked about the fact that the introns are cut out and the exons are expressed, but we can have different ways of editing that messenger RNA. We also have different enhancers and other transcription factors that can actually fold the DNA so that transcription is more likely to occur, and it enhances the attachment of the RNA polymerase. And we're going to talk about that later. That's an important way that we can control. So in a eukaryotic gene control model, here we have our promoter region. Remember, that's where the RNA polymerase is going to sit. And this is the DNA, so the RNA polymerase is sitting on here. And then downstream, we would have our exons and introns. We have a poly A signal sequence here, and then a termination region that we have here that is going to tell the RNA polymerase to stop transcribing. Our primary or pre-messenger RNA transcript is going to have a 5' area here and our 3' area here. Our polyadenylate signal, which we transcribed from the signal sequence here, is on here. And now we're going to have some RNA processing. So we have our spliceosomes, remember, that cut out our introns. We have a 5' cap that is put on that helps to stabilize and get the messenger RNA out. We have our poly A tail that is going to try to prevent the messenger RNA from being degraded as soon as it goes out into the cytoplasm. Upstream from where the promoter sits on the DNA, we have some enhancers that can sit on these DNA sections and can actually help fold the DNA up and make it more likely to be transcribed. So remember, the Tata box is a characteristic that is seen in many eukaryotic cells in the promoter region that helps to guide all these proteins that are going to be part of our transcription initiation complex. And we can see here that activators in the cell can actually bond to these enhancers, and we have some that are distal and some that are proximal to the promoter. That's not really such a big deal. But anyway, these activators can bond, and together with these proteins called DNA bending proteins, what happens is the DNA molecule actually bends down, and that helps to initiate this transcription complex. And our RNA polymerase is going to fit on here, and now we have RNA synthesis, we have our transcription. So this would be an example of transcriptional control in eukaryotic cells. So remember when I was talking earlier about how cells are different because of the different proteins that they make? Well, here's a great example of cell differentiation. So here we have a liver cell, and one of the proteins that a liver cell makes is something called albumin. Albumin is necessary in our blood because it is something that helps to control the osmotic pressure in our cells and in our fluids. So in our lens of our eye, one of the proteins that we need is a protein called crystallin. So our liver cells have the gene for crystallin, but they don't make crystallin. And our lens cells have the gene for albumin, but they don't make albumin. So how did that happen? Well, we know that it happens by different activator molecules that are in those cells, that are released in those cells. And so they could be a result of some type of signal transduction pathway, but we have these activators that are going to bind to these enhancer regions, and that's going to help the transcription and then later the translation of the gene for albumin. The crystallin gene is not expressed because these activators do not work on that particular gene. Similarly, in the lens cell, we have different activators that are present. Those do not bind to the enhancers in the lens cell for albumin, but they do for the enhancers upstream from the crystallin gene. So this is an example of how different cell types have different types of transcription and then translation to make cells differentiate, to make different proteins. Another way that cells can control protein synthesis is by altering the post-transcriptional processing. In other words, they can splice the messenger RNA in different ways. Okay, so there is a protein called troponin, and troponin has a little bit of different varieties in that. There are different types of troponin protein that the cell can make. So in one primary RNA transcript, you might have certain RNA splicing that allow these sections of the DNA to make it into the final copy of the messenger RNA that is then going to be translated. And I can see that different exons made it into the final messenger RNA transcript after a different type of splicing. And so this is an example of how we can get different protein products from the same gene in the DNA, but just by splicing differently and having different exons that are left in there. Another example of post-translational editing, all right, so this is an example of after translation has already happened, we can control what proteins get chopped up and recycled and which ones stay around. So if there is a protein that the cell wants to get rid of, now it's already transcribed and translated it, but maybe it doesn't want it around anymore, we know that there are these proteins called ubiquitin. And when those bind to the protein, they help to guide this complex. Remember, this is a protein that's destined to be destroyed into a big protein barrel thing called a proteasome. And so what happens is this ubiquinated protein, because it has the ubiquitin on it, is now being recycled. It's in the trash can, and it's being chopped up by degradatory enzymes, and now these protein fragments can be recycled and used elsewhere in the cell. So this is a great review slide of all the different places in eukaryotes that gene expression can be regulated. And I would suggest that you go back through and make sure that you understand all these different areas, different levels where it can be regulated. One of the things that I want to talk about very quickly is the explosion of information that we have been learning about RNA. It is looking like it really is an RNA world. And that all these different kinds of RNA are helping to control gene expression. And so we have one big type of area called non-coding RNA, and then we have coding RNA. An example of coding RNA is our messenger RNA. So there is a great tutorial on the Howard Hughes Medical Institute site, and I put the link down here. If you want to pause the video, for those of you in my class that actually have this slide, that link works and you can go to that tutorial. For those of you that are watching this that don't have the slide presentation, you can just copy that link and put it in your browser and go through that tutorial on different types of RNA. I highly recommend it. So one classification of RNA is called non-coding or ncRNA. And this is going to be RNA that is not translated into a polypeptide. So examples of non-coding RNA that we already know about are rRNA, so that's the type of RNA that makes up ribosomes, and transfer RNAs, which we know are the little shuttles that bring the amino acid to the correct spot on the messenger RNA. But we have all these different new types of non-coding RNA that we have just discovered. MicroRNA, small nuclear RNA, interference RNAs. We have small interfering RNAs. We have Pee-wee associated RNAs. There is a great 4-minute video that Khan Academy makes on non-coding RNAs. If you just Google Khan Academy non-coding RNAs, there's a great 4-minute tutorial on that. I just wanted to take just a minute to talk about miRNA because it's very interesting. So one of the things that we have learned about miRNA or microRNA is that microRNA can actually bond to a piece of messenger RNA and can either and can actually block it from being translated into a protein. So the way that this works is that if you have a piece of messenger RNA that is transcribed, it actually makes these little hairpins. They're actually hydrogen bonds that hold the different pieces. Remember, RNA is single-stranded and it bonds together. And then there's a little protein called dicer that cuts off the ends. And then one end of the miRNA is degraded. So now I have a single strand of miRNA. This is not going to be translated. It actually associates itself with the protein complex, and then what it does is it will complementary base pair to a stretch of messenger RNA. If almost all of the bases on the miRNA complementary base pair with the messenger RNA, then that messenger RNA is slated to be destroyed. If only a few of the bases on the miRNA complementary base pair with the messenger RNA strand, then the translation is just blocked. And so this is another level of control that we have just learned about in just the recent past about how miRNA can control gene expression into proteins. And of course, spliceosomes are another example of non-coding RNA. So this is not going to be translated into a protein, but it's going to be used as a ribozyme. So here we have these small nuclear RNAs that are part of this big protein RNA complex called a spliceosome. And this, of course, we know is what cuts out the introns on the pre-RNA before it's been edited. And we can do differential splicing of that to get different transcripts. I just want to end the screencast to just talk about the amazing things that we have found out lately about different types of RNA and different types of diseases. I would love it if you could just take a minute to just freeze this screencast right now and take a look at some of these and just go out there and see what you can find out about this. It's really amazing stuff. So I hope that's been helpful, and I will see you in class.